Well, good morning. Back in 2012, the State of Redwoods League was nice enough to give me a grant to study population genetics of two species, two common understory species of the redwood forest. One was western sword fern, and the other was redwood sorrel. The western sword fern, I think because it sporulates and spores just blow everywhere, there was really no, it was pretty homogeneous genetically throughout the redwood region. But I found some interesting things with redwood sorrel, and that's what I want to talk about today.
Excuse me. Could you stay behind the podium?
Yeah.
Sorry.
Okay.
Okay, for future speakers, we are recording, so please use the microphone.
Oh, I forgot about that. I'm used to teaching and walking several miles. Okay. I found evidence for two very distinct genetic lineages of redwood sorrel in California in the Redwood region. And what I want to show you the evidence for that and also throw out some ideas about why that occurred, including maybe an ancestral population during the Pleistocene that during the glacial periods split up into different glacial refugia where they diverged substantially. And now they're thinking about coming back together again. And actually I know in one plant they did. Oh, excuse me, here I go. Distribution goes from British Columbia, Vancouver Island, down to near the end of the Redwood Range. There's a few outliers. I'm not sure if those are real or planted in someone's garden. This is the Oxalis oregana distribution in California. You can see it's essentially the same as the Redwood distribution, which is there in red. I had, a few years ago, about 5 or 6 years ago, I had done a range-wide genetic study of redwoods, and I just kind of went back to some of those sites and took samples of Oxalis. And so I looked at 7 different populations that go from Monterey County all to Del Norte County, and I took 10 samples of each, and I looked at the chloroplast, several chloroplast loci. I'm going to be talking about 2 of these, the one on the top and the one on the bottom, the TurnQ RPS16 and the PSBJ PET A. These are intergenic sequences. These are not gene sequences, but they're just the spacers in between the genes where they tend to mutate at a faster rate. I'm not going to talk about the one in the middle, but the results were essentially the same as the other 2. These are in an area called the large single copy region, which takes up more than 50% of the entire genome. Now, this is the PSBJ PET A haplotypes. I found 6 haplotypes. Now, the strange thing, which drove me crazy for 2 years, and it just finally came together the last 2 months, thankfully. 3 of these haplotypes are what you would expect from a haploid chloroplast. You expect one sequence. And I found 3 haplotypes that had single nice sequence. You can see there's a base substitution in one, and there's and then that area of 9 bases there. There's a base substitution there as well. But then I found 3 other haplotypes that I'm calling mixed sequence haplotypes. It's as if there were 2 amplified, 2 PCR products that I was sequencing. And I was getting 2 sequences at a time for each individual. And I was able to tease the sequences of each of those out, and that's why there are 2 sequences for each of those. Notice that they're fairly related. You have a 9-base deletion in some of them and a few substitutions here and there. And a little bit later I'll talk about why I think that might have happened that way. But anyway, this is an example. Well, it doesn't show it very well, but haplotype A is a single sequence. Haplotype F is a mixed sequence. There's a base substitution pointed out there. But about halfway through the sequence, for the mixed sequence, there's a point where the sequence is wonderful, and then you get to a point where you start getting the double sequence. Okay. And that's, if you stare at that long enough, you'll realize that that's because in one PCR product there was a 9 base pair insertion, but it wasn't there in the other, and that caused everything to be offset. If you look at the distribution of haplotypes, I have the mixed haplotypes are in the orange and red shades, and the single haplotypes, single sequence haplotypes are in the blue shades. And there is a very distinct phylogeographic distribution. Just an abrupt change between the two types of haplotypes. Now, I then went to the next spacer, the next locus, and I found one single sequence haplotype, haplotype 1, and then I found 3 mixed sequence haplotypes. And you can see here with the single sequence haplotype, you get one nice base at that position, but you look at the same position in the other mixed sequence haplotypes, and you get an A and a T together. So again, it's like there are 2 templates being sequenced. But if you look at the distribution, that looks familiar, doesn't it? Looks just like the other one, where the mixed sequence haplotypes range from, this is Humboldt Redwoods, their HU, all the way down to Pfeiffer Big Sur in Monterey County. All of those had mixed sequence haplotypes. Only in Prairie Creek and Jed Smith did you see the single sequence haplotypes. Notice in both loci there are a couple of individuals. This pie chart represents 10 individuals. And there are 2 individuals in Jed Smith that do have a mixed sequence haplotype. So you see the mixed in the northern population, but you don't see the single haplotypes in the southern populations. Now, what could be causing this mixed sequence haplotypes? Well, there's lots of possible reasons. The first 2, I did everything in the PCR handbook. Everything I've learned over the past 100 years that I've been alive to make sure it was not a PCR artifact, because that's the first thing you think when you get double sequences. You think it's either a PCR artifact, a secondary structure, or maybe a sequencing artifact. But why would you get an artifact only in some samples but not others in this geographic distribution that I'm getting? Secondly, I said, oh my God, did I contaminate my samples? And so I went back and I re-isolated DNA from all the samples again. Exactly the same results. So I'm convinced that what's going on is not my fault. I'm blaming nature on this. There are two reasons. One could be simply, and this happens in chloroplasts, you can have a locus that just duplicates within the chloroplast, inserts into another place right in the same circular molecule, and then diverges, mutates. And so you could amplify 2 on the same chloroplast molecule. And that's possible, but to have that occur in 3 loci spread all over the large single copy sequence, plus I have evidence that there's a 4th locus doing the same thing. And I have not found any plants in the literature that have so many duplicated loci like that. The explanation I'm leaning towards is the last one, where we have chimerism. That's where you can have, within the same leaf, you can have cells that have different types of genomes. And if there is, say, a wild type chloroplast in some of the leaf cells, but a mutated chloroplast in some of the others, then if you isolate DNA from that whole leaf, then yes, you're going to get 2 different chloroplast sequences. Now, it turns out that there are many species of Oxalis that are chimeric. A nice visual chimeric example is leaf variegation. Because in some cells, you have chloroplasts that have been mutated to the point where you're not making chlorophyll anymore, and so those areas are white. And But I'm thinking that there's a possibility that I'm getting mutations in a chloroplast in some cells, but it's not enough of a mutation to cause a chlorophyll problem, and so the tissue is still green, so I cannot tell which cells might have the different chloroplasts in them. Okay. Now, this has an implication because chimerism is not propagated sexually. It can only be propagated vegetatively. So this implies, perhaps, that the vast majority of propagation in the southern lineage is vegetative. And we all know that Oxalis does a very good job of propagating through rhizomes. Also in redwoods, it's thought that in the more southern redwoods, the farther you get to the south, the more clonal propagation you have and less sexual propagation. Less moisture, higher temperatures, it's just harder for seeds to get going. So this is all kind of, in theory, it's making sense. I haven't proven it yet. Now, since the chloroplast work was just driving me absolutely crazy, and I knew that there were going to be some people out there who would not believe that it wasn't my fault, I decided, well, it would be really nice to have a nuclear marker to confirm this. Now, I didn't have the time or the resources to throw together microsatellite markers, which would have been nice. So I went to an area of the nuclear genome that it still has a relatively high mutation rate, and that's the internal transcribed spacers, ITS1 and ITS2, that you find between ribosomal DNA genes. Now, most of the time, when you look at these regions in individuals within a species, the sequence will be the same within a species. However, in some old work I'd done with spine flowers back in the Bay Area, I did find there were some varieties of spine flowers that had minor differences in the ITS region. So I said, well, I have nothing to lose. I'm desperate. I have the primers in the freezer. Let's go for it. And I'm glad I did. What I found 6 very highly related genotypes in what I'm calling the southern lineage. These would be individuals in Humboldt South. And you notice that they all differ just at one position. Okay? Those little dots indicate that they're all the same as that S1 genotype. Now, the S1 genotype, I'm assuming that both alleles are that one sequence. S3, for example, one allele would be the top sequence, and then the other allele would have an A at position 136, so that would be a heterozygote. But if you look at what I'm calling the northern sequence, you can see that there are, if you count them up, there are 12 differences between the northern sequence and the southern sequence. And does this look familiar? Again, the southern genotypes, S1 through S6, are in the orangey reddish colorations, and the northern genotypes are at the top. Okay. Now, notice that I have one red and blue. I did find one individual that appeared to be a hybrid between these two, okay, which indicates to me there might be a little hanky-panky going on there between the two. Okay, so you put them all together, all 3 loci, and that's what you get. So I think that's pretty good evidence that we've got a genetic break going on somewhere in Humboldt County, right there. And maybe some migration, because you see red and oranges up at Jed Smith. There might be some migrations of the southern stuff up to the north and the like. Now, why is that? Well, it turns out that the Klamath-Siskiyou region is an area in central Oregon down into northern California, and you notice the southwestern boundary, just this side of the Klamath River, that runs through Del Norte County and Humboldt County. Now, the Klamath-Siskiyou region was an area of high genetic diversity, and it was also a known glacial refugia for some species, such as this ride species. It's a wild current. Notice that some DNA, cpDNA, chloroplast DNA haplotypes, black, are there, which correspond to the Klamath-Siskiyou region, and those that probably derive from a different glacial refugia are in the white circles. So you can see that this Klamath-Siskiyou region could be a glacial refugia. Now, if you map the Klamath-Siskiyou region against my northern three populations, you find that Jed Smith is within the Klamath-Siskiyou region. Prairie Creek is right on the edge. Prairie Creek is in the Redwood Creek watershed, not in the Klamath River watershed, but it's only just a few miles. And the elevation between the two is like 1,000 feet, a little over 1,000 feet. So I think there's a possible pathway between those. Okay. But I think that the mountains that come more southeast, those get higher, 3,000, 4,000 feet, and I think perhaps you might have a barrier there, right, between the Redwood Creek and the Klamath River watershed. So that could be the barrier to migration. So I think what's going on is that you have a southern refugium in the south there, where a population hung out, and then you have a northern refugium either in the Klamath-Siskiyou region or further north. And if it was further north, post-glacially has since migrated down south. Now, finally, an interesting thing here, I did the phylogenetic tree. It turns out the northern genotype that I have is exactly the same as a genotype from an accession in Oregon that I got off the gene bank. Exactly the same. So I think these two are highly related, and so there must have been a bottleneck that reduced it down to this one sequence, and they since spread very rapidly. In the south, they had more room, perhaps, greater populations to diverge. And also, you notice that the northern type is more related to a different species than it is to the southern lineage, which indicates that these have been separated a long time. Which begs the question, are these lineages different enough to be considered subspecies? Well, they are geographically separate, which you need. They are genetically distinct. They are capable of hybridization. But this, I don't know if they're morphologically distinct or not. And that's what I'm going to have to try to do for my future research. I'd like to look at things north of California. I'd like to see what that dividing line is a little bit better. I want to test my chimerism hypothesis and then do a morphological study on this. And then maybe check other species to see if they have the same type of pattern. So I want to thank, say, the Redwoods League for the bulk of the financial support. California State Parks, we're issuing a permit, University of Maine DNA sequencing facility, and they do sequence wicked good. And finally, when the Save the Redwoods League money ran out, I want to thank my wife, Bonnie, for picking up the slack and cutting checks to the sequencing facility. Okay, so thank you very much.